Hello, everyone. So today we have a very welcome guest all the way from the USA, Dr. Michael Love. Mike is an assistant professor in the Department of Biostatistics and Department of Genetics at the University of North Carolina at Chapel Hill. And his lab uses statistical models to infer biologically meaningful patterns in high-dimensional datasets, as well as developing open source R packages. I first came across Michael's work many years ago, having used the extremely popular DSEC2 package for RNA-seq differential expression analysis. However, Mike's current research interests include a variety of topics, such as integration of multi-omics data with MR, RNA splicing, allelic expression, as well as application in neuropsychiatric and breast cancer genomics. So today's talk is going to focus on MRLocus, which is a new method leveraging allelic heterogeneity of cis-eQTL signals for a given candidate gene to prioritize genes causally involved in different complex traits. And yeah, over to you, Mike.
Thanks so much, and thanks for having me today. So I'll pull up my slides. Everything look okay? Okay, great. So today we'll talk about estimation of gene to trait effects via eQTL and GWAS summary data. I haven't updated the date here, sorry about that. The slides here are, there's a bit.ly link if you want to follow these later. So it's just bit.ly MRLocus. And yeah, I'd be happy to take questions during the chat, like whatever's easiest. You could during the talk, you could just put them in the chat, for example. So first off, I want to acknowledge the first authors and other folks who worked on this. So we had two first authors on this work, Dr. Anshi Zhu, who was a PhD from UNC Biostatistics, who's now, she was at 23andMe. She just recently moved over to Genentech, and Dr. Nana Matoba, who's at UNC Genetics and Neuroscience. This work was in collaboration with Dr. Jason Stein in the neuroscience department at UNC. And I'm just happy to mention that it's International Women's Day. So I hope everybody has a happy International Women's Day. Also, it's a lot of great co-authors on this work as well. So Yun Lee, who's in UNC Genetics, helped immensely, giving us a lot of insight into GWAS and also mediation. Amanda Tapia worked on the project from Yun Lee's group, and then also Emily Wilson worked on this project. She's now in Karen Mulkey's group at UNC Genetics, where she studies cardiometabolic disorders and mediation through tissues like adipose and liver and muscle. And she's actually gonna probably be doing some kind of mediation analysis or Mendelian randomization analysis very soon with metabolic traits. So a diagram, a sketch of our causal DAG that we're trying to use here. So we're performing 2-sample Mendelian randomization. Obviously inspired by a lot of the work from scientists before, including a lot of work from the MRC. Just to start out, we're interested in selecting cis genetic variation and using gene expression as the mediator. And we purposefully select 2 or more what we'll call nearly independent variants cis to the gene. So we're not attempting to do this with a single instrument, and we'll kind of, our way of tackling the potential pleiotropy, which could confound the arrow of interest, which is gene expression onto trait, will argue that given the cis relation to the gene, this causal edge is less of a concern as we aggregate signals. Of course, other methods have explicitly handled pleiotropy, and we're interested in potentially developing, more robust inference by explicitly handling pleiotropy, but we we don't really use that now. Right now, we're relying on multiple signals to have confidence in the expression to trait causal arrow. I think there's a great concern about weak instrument effects. Weak instrument effect size, especially as N is growing quite large for the QTL experiments. So we looked at, for example, QTL sample size of 30,000 eQTL gen. And this is something I think we definitely need more work on this arrow, because you'll accrue a lot of instruments if you use kind of heuristic cutoffs. And then, yeah, so the orange comment boxes are kind of, areas of concern that I'm just highlighting at the very beginning of the talk. And the yellow arrow is kind of an interesting, more of an interesting question about how to go about choosing tissue, or perhaps modeling across multiple tissues. So it really matters if we could have loss of power if we don't choose the right tissue or the right context. Additionally, today I'm going to be talking about modeling a single gene. Of course, there could be multiple genes acting in a locus in concert, and that's an interesting avenue that others have pursued more than us. We're modeling the total expression level, but perhaps we should be looking at splicing or instead at post-transcriptional, so at the level of protein abundance. That would be very interesting, and we're interested in moving in that direction, but we don't have that today. So we're relying on multiple instruments in a locus, and these are eQTL, what I'll call signals, and that's kind of following the language of a method called PTWAS. Let me just make sure I have my time. Okay, so here I've colored 3 signals using LD clumping. And you can, I've highlighted the kind of the sentinel variant in an eQTL for liver gene expression for the expression level of lipC. And if you at the bottom, if you superimpose GWAS for high density lipoproteins, you can see that some of these have a paired signal in the GWAS. And I guess this is probably familiar for this audience, but this is kind of my introduction to the idea that multiple signals in a locus could help you better estimate the effect of a gene on a downstream trait. And even this purple signal, although it's not as large, it's not as intense in terms of the log minus log 10 p-values, it also has significance. It's just not as extremely significant as the green or the red signal. There's other ways to disentangle signals, and we're very interested in moving from LD clumping based on reference panels, which can be imperfect, to performing conditional analysis on the QTL study. That might be a better way to disentangle overlapping signals. So moving right into conditional analysis, in particular in collaboration with Karen Mulkey's group, we've kind of moved towards our current applications where we, if we have the data, the QTL data on hand, we can perform conditional analysis by conditioning on top signals to highlight other signals that are superimposed. And so here's a case where there's 2 signals in the eQTL that are kind of on top of each other. After conditioning on the purple variant, the green signal is easier to isolate. And actually, in this case, it's likely the green signal set of variants, which is associated with waist hip ratio in this case. So this is definitely ongoing work. I mean, this is a publication kind of showing the possibilities of using conditional analysis and exploring allelic heterogeneity. And we're definitely doing ongoing work here. So if we focus on genes that display allelic heterogeneity, that's obviously a subset of all possible genes mediating traits. So we're getting a reduction in power just by assuming that we can identify multiple signals. So how many could there possibly be? I think for me, the gold standard is conditional analysis to kind of identify these rather than approximate conditional using summary statistics or LD clumping. And so just, these numbers rely on the sample size of the study and on the tissue and many other things. But for example, some numbers from recent publications were 35% of E genes. Here, a sample size, a blood eQTL study with nearly 5,000 donors. Other estimates from GTEx put the range across different tissues at 4 to 23%. There's a high correlation between the number of loci with conditional signal, multiple signals, and the sample size. So that's kind of promising that as we get larger and larger samples, we will be able to identify secondary and tertiary signals. There are of course other groups that are interested in this type of analysis. This kind of like, maybe some have called it cis-MR. And so an example of a recent paper looking at neonatal genetics of gene expression and autoimmune or allergic disease risk. They perform eQTL in myeloid cells and CD4 positive T cells with a sample size of 150. They found this many E genes. Then they kind of did a subset of those that have 3 or more genetic instruments. So you get this with a sample size of 150, you're going to get a big reduction, but then you can, I think, really pursue those genes with more confidence. And so here are some mediated effects on disease risk in terms of log odds per standard deviation of gene expression. And so I'm really most interested, or the group that developed this method, we're most interested in this, in putting effect sizes in meaningful units. So I think this is a great unit that we can try to estimate from a 2-sample MR and compare across different methods. And also, in particular, getting the confidence intervals correct. So that was kind of the big focus of our work, was not just null hypothesis testing, but also focusing on the effect size and on its uncertainty. So I mentioned, getting the tissue right is very important, and so we've used just standard TWAS as a screening approach, because I think for our methods we kind of require TWAS significance in order for it to possibly be significant in our methods. So we'll kind of be a specificity filter on top of TWAS in the same way methods like focus might be, kind of improving the specificity once you've cast a wide net with TWAS. And so there are ways to kind of identify it with a screening step, like which tissue and which gene might be good candidates to follow up with this cis-MR. There's another method I want to highlight that kind of came out recent, like while we were working on MRLocus before we'd put our paper up, and it's called PTWAS. And it did kind of go in a similar direction that we were hoping to. So we, instead of, we highlighted it and we compared to it in our paper. And we also, instead of like coming up with new terminology, we just adopted a lot of the terminology from that paper. So we refer to a signal cluster as a set of SNPs in LD that are deemed to represent the same underlying causal eQTL, but it's not necessarily identifiable based on the association data. So we definitely compared to this one and other methods in the space. So jumping into our steps, and these are the steps that we used in the paper. We used LD clumping to identify these, what we called like signal clusters, which are nearly independent. They're not independent. They do have some across cluster correlation. We also are interested in doing this with conditional analysis. So that might be a better way to identify, because it relies less on the hope that the LD reference panel is appropriate. So these were our parameters for LD clumping with Plink. R squared of greater than 0.1 puts variance in a cluster, and we're clumping with a raw p-value, eQTL p-value of 0.001. We looked at lowering this to require stronger instruments, but we showed that it did not improve accuracy of the gene to trait effect. So that's, of course, this is a heuristic. This is, you know, a threshold, and so it induces all kinds of downstream problems if that's not the correct threshold. And I think that's actually the wrong threshold when you get to like 30,000 samples in the QTL study. So here, you know, imagine we have these three signal clusters, which are identified just by looking at the QTL study, and we would then rank them by their effect size. These will then go into, we will take summary statistics both from QTL and GWAS and the intra-clump LD matrices, and this is basically the input to our method. So this is kind of upstream, and then this is the type of data that's being supplied to our method. And we also, so summary statistics would be effect sizes and standard errors. We take care of allele swapping internally. So, you know, the GWAS and the eQTL study might not be on the same allele as well. Also, like we need to, because we're modeling effect sizes, we need to make sure that within a clump, within a signal cluster, we swap the alleles so that the LD matrices, we have positive correlations as much as possible. We collapse, you know, very highly correlated SNPs. I think it's 0.95 R squared. So those just induce problems in modeling. So we pick out one candidate SNP among very highly correlated SNPs. We flip alleles so that all instruments are positively affecting gene expression. That just really just makes the plots easier to visualize because you've got all the data on the right side of the plot. We then perform, within signal cluster, we perform co-localization of eQTL and GWAS. We have a method for this, but we also found that other existing co-localization methods worked as well. So we either have a Bayesian model that we attempted, and we also showed that eCaviar worked as well in that step. We then trim these clusters to ensure that the R squared across clusters of sentinel variance is less than 0.05. So at this point of the top candidate for co-localization. So we're trying to make sure that this does not get too high because that obviously impairs inference in the next step. We pass along the top eQTL. So we're picking based on the posterior effect size for the eQTL study because we want all candidate instruments. And then the last step is estimating the gene to trait slope. And importantly, or I think, you know, one way that we distinguish ourselves, I think, was that we are counting for error in both directions. So we're not just accounting for error in the GWAS or the downstream trait, but also in the eQTL effect size. So this was we have a Bayesian model for co-localization. It follows some similar work previously. So actually it looks a lot like the eCaviar model. So we assume that the estimated coefficients follow a normal distribution with the LD matrix multiplied by the true coefficients and given our estimated standard error. So in this simulation, if we have a true variant here among these 9, for, you know, this is just a small example to show a demo of how it works. Obviously the LD matrix induces these marginal associations of nearby SNPs in LD, both for QTL and GWAS. And Then we have a Bayesian priors for the true betas. And here we used a, what's called a horseshoe prior which has this lambda I is a local shrinkage, and then tau is a global shrinkage parameter. And so in order to do this, we have to scale the betas upstream, and then we unscale them afterward. But we found this actually worked quite well in, it induces sparsity. It favors sparse solutions. It doesn't send the other, the associated by LD variance to 0, but it helps to identify, it helps to pluck out the the likely true variant. And if in the case that there's not co-localization, we found that it was better than existing methods at identifying the causal QTL. So we found that other methods that rely on p-values here were strongly influenced by the GWAS, so they would end up kind of favoring GWAS, whereas we end up with posterior effect sizes for both, and then we would pick based on whichever has the larger posterior effect size for the QTL study, because we want instruments. So I know that was really quick. I'd be happy to answer more about that later. Then the last step is then taking candidates per signal cluster and then performing estimation of slope. So here we have, again, the estimated coefficient, and we will use the posterior effect size from the previous step as our beta hat. And we assume that follows a normal distribution according to some true effect size, and it has standard error. So there's uncertainty both on the effect size of the instrument in the QTL study, as well as in the downstream GWAS. So here, sorry, I should have said A would be the potential mediator, so like gene expression, and B is downstream trait like GWAS trait. We then assume some distribution for the effect sizes on the x-axis and a distribution on the y-axis. Here we just have an alpha slope, so that's our parameter of interest, times the effect sizes on the x-axis. We could here add an intercept, but we haven't done that yet. So that would be potentially accounting for horizontal pleiotropy, but in this first round we didn't put an intercept here. And then we have a couple more parameters. I think I'll be helped out by adding some extra diagram here. So I guess the things to highlight are we do account for uncertainty in the x-axis. I think that was a little bit unique. I'm not, I know some groups have done this as well, but it's not common to take into account the uncertainty on the x-axis. And then our parameters of interest here is alpha, like I mentioned, the slope, and also the dispersion. So how each signal cluster varies along the trend. This sigma is kind of an interesting parameter in itself. And so a diagram of these parameters on the right-hand side would be, the effect sizes on the QTL on the x-axis and on the GWAS on the y-axis. And we have so the gene to trait slope is this heavy blue line, alpha hat. We have an interval, like a credible interval on the slope. That's this dashed, the cone with the dashed lines. And then we also have a band, which makes use of our estimated sigma. So this kind of thin band is, and so that's, I think, an important distinction, differing their inference on the slope and the cone of uncertainty about the slope, and then the dispersion of the individual signal clusters from the slope. And just to show an example of, so alpha is here and sigma is here. An example of how you could have like differing uncertainty levels. On the left is the plot I just showed you. In the middle would be, we're confident about the mediation effect, right? So the cone is, we have some uncertainty, but it's not 0. The 80% band, 80% interval for the slope is not overlapping 0. But there is high dispersion across individual signal clusters. So that increases this light blue band, but we're still have confidence about the heavy blue line. And then on the other side would be, we're no longer confident that there's a mediating effect because the dashed lines cross over 0. We evaluated this method using 3 scenarios, and here I'm borrowing diagrams from a nice paper that came out also while we were working on this called MRLink. So MRLink is not only for, it cannot only run with summary statistics. So that was like, we didn't evaluate against MRLink, although it's definitely in the same space. So here I'm using their diagrams to show the kind of simulations that we did. So the concern would be that you have SNP-A, which affects your exposure of interest, but it's with this chain, it's showing LD. So it's in correlation in population with SNP-B, which might be affecting some unobserved exposure. So in what I'll call the simple case, which is the alternative hypothesis, that SNP-A affects the observed exposure and that affects the outcome. That's the very easy case. And this arrow here would be the arrow that we're interested in. We also simulated horizontal pleiotropy, which is kind of the diagram on the left, that you have both a direct effect through the exposure of interest, and then you also have within the same locus other SNPs acting through a different pathway on the outcome, not through your exposure of interest. And this then is a problem, and it could impair inference of that causal arrow. And then finally, we have null simulations where we have SNPs that are linked to the exposure. So our eQTL signals, those are in LD with an unobserved exposure, which affects the outcome. And there's no mediated effect through the exposure of interest. So I'll call these the simple alternative, horizontal pleiotropy, and null simulations. We used an existing framework, so we didn't create a new simulator. We just used something called TWAS sim from Mancuso lab. We simulated eQTL and GWAS using 1,000 genomes. I think they're probably better LD panels that we could use now, larger ones, and also interested in extending this to kind of explore non-European ancestry groups. But this was our first attempt. And our sample sizes were kept at the default values for TWAS sim. So the default there, I think GWAS inspired, was about 500. That's for a large GTEx, a tissue with a large sample size from GTEx, and 100,000 for the GWAS. And we ran with the setting that 1% of SNPs in a locus were associated with gene expression. So this is an example of all the simulations that we ran. So here, this is the heritability of gene expression. So we'll call that H2G, and we varied it from the default value of 10% up to a high value, 20%, and then down to 5%. And then there's just to highlight, there's a couple other scenarios here. There's the horizontal pleiotropy. There's a sample size increase for the QTL study. And then over here, there's 3 null scenarios. So this is the expression heritability. And then we also varied the mediated heritability through expression level. So the default value is 1%. We moved it down to 0.5% and 0.001%. Sorry, 0.001, a tenth of a percent. And then null, obviously, is 0% expression mediated trait heritability. So, these numbers kind of seem like they're drawn out of the blue. So, these can be informed by recent work. So, for example, the MESC paper tried to quantify genetic effects on disease by assayed expression levels, and they are estimating that 11% of heritability was mediated by expression heritability across all genes. And this is obviously, this is an average, so it's going to go up, it's going to go down for different traits. And this is across all genes. So the amount that we can expect for a single gene has to be much lower, likely much lower than this. But you can look up the varying mediated heritability, and this is the ratio actually of mediated heritability over trait heritability. But you can look up these values, and I think it makes sense. You can somehow these make sense, like especially when you have traits that are closer to the underlying biology, they're becoming kind of like endophenotypes. You can get higher ratio here. Yeah, so it's just putting some numbers to those simulated values. But I think just to go back, like 1% is obviously high. The idea that 1% of trait heritability is mediated through a single gene is a high value. Okay, but and just to say, the denominator here can be large. So these were the methods we compared to. The first two are kind of the first one is just using inverse variant weighted 2-sample MR using all SNPs. Just throw all SNPs into a IVW model. The second one is kind of an oracle. So this is using the true causal SNPs. So just performing IVW, inverse variance weighted 2-sample MR, knowing from the simulation the SNPs that are mediating. I think, sorry, it's knowing the SNPs that are instruments. So not all of them have to be mediating in the null or the horizontal pleiotropy simulation. And then these are kind of competitor methods, so TWMR, PTWAS, and this is the upstream method for identifying independent signals or nearly independent signals in the case of clumping. Our method, both for co-localization and slope estimation, our method with eCaviar for co-localization, and we're just using the slope estimation model. And then two other methods, LD-MR Egger and PMR summary Egger. I know there's probably even more methods. This was kind of at the time when we began writing, these were the best. These were the ones that we were aware of. So I'm going to start with the results for the null simulation, just because that kind of sets the stage, I think, in an important way. Along these columns are different methods, and then the rows indicate different expression heritability. So 5%, 10%, 20%. Within each panel, there are 20 simulations, and the point is the effect size estimate with the 80% confidence interval or credible interval, and then it's red if it does not contain the true value here, which is 0, and then it's black if it does contain. And then shown up at the top left is the percent of it's the percent of intervals that cover the true value. So we would want this to be near 80%. And then at the bottom is an estimate of the error on so the average error of the effect size. So we can look at the error. We want the error to be small, and we want the coverage, the number on the top, to be high. And I think just kind of what motivated our focus on the instrument effect size standard error is that the existing methods, I think the intervals are too small. And my hypothesis is that it's because it's not taking into account the standard error of the QTL effect sizes. So here we're dropping well below 80%, whereas our methods, while not always hitting the target, like here we're a little bit low, 50 instead of 80%. We have I think more appropriately sized here credible intervals. Here's an example of one of the results in the case of the alternative hypothesis. So where the gene does mediate trait heritability, and these two methods that we propose on this is just running an existing simulation framework. We didn't modify the framework at all for this. We found that we had good relative mean absolute error on estimating the slope. So here the slope, the true slope is on the x-axis and the estimate is on the y-axis. And of course, we're not doing as good as not having knowledge of the true instruments and then performing IVW. But we're doing fairly good, I think. So this is the relative mean absolute error relative to the true slope. And then the other is just the mean absolute error. So what you see on the y-axis. So that was just focusing on the effect estimation, effect size estimation, and the error. Here's looking also at the credible intervals or confidence intervals. And so the causal, what we call causal, which was this oracle method, also has too small of confidence intervals. And that's because, in my opinion, it's not taking into account the substantial uncertainty on the effect size in the x direction, x-axis, like the QTL uncertainty. So it does very well in centering on the true slope, but the intervals are too narrow. And then here we have, PTWAS is doing a lot better than other methods. And then here our methods have much larger intervals, and they're actually covering the effect size. And also I should mention, right, so that even though the intervals are larger, if you look at the bottom, we have the 0 axis. So they're mostly not overlapping 0. So we wouldn't, it's not that because we have larger intervals, we would be losing sensitivity. This is a summary over looking across many different variations of the simulation. So on the top, higher expression heritability, and then within each panel, low to high expression mediated trait heritability, and we're on the y-axis is the relative mean absolute error, and so we're doing pretty well. We're not as well as the oracle method in terms of error, but that's expected. And then I just want to highlight, like, one way to evaluate simulations is to look at all the methods except the one that of the authors, and if you use that rule, then PTWAS was winning the simulation. So if you think that we are biased and therefore you shouldn't look at the red lines, you could focus on the blue line and show that it's doing pretty well. We also simulated cases of horizontal pleiotropy. So into a locus, we inserted also large effects, SNPs that are associated only with the trait, not mediated through gene expression. So this is kind of just an example of what that looks like. Adding in here 3 signals. Maybe this is a lot of signals in a single locus. And in that case, we still were able to have good coverage of the intervals. Although adding in horizontal pleiotropy signals definitely increases everyone's error rate. Let me see. I might speed up a little bit so I stay on times. And also I've shared the slide. So I want to move into, there's actually some, I think, interesting problems with current methods that I'm going to, so I'm going to jump ahead to like application to real data sets. And so we looked at artery QTL and liver QTL paired up with coronary artery disease and HDL and LDL. And this is just a summary of each method and its effect size estimate and intervals across different cases from literature that we kind of expected to see results. And we're generally, in the same range as existing methods. So we're not kind of totally coming up with a totally different effect size. And we do have these larger intervals. So, it's hard to market your method as having larger credible intervals, but I think it's actually pretty important to accurately pass along the uncertainty from this in this type of analysis. And I want to highlight down here, this is LIPC as measured in liver and its effect on HDL, and the effect size is negative. And that's actually expected given our understanding of how LIPC works, right? So it would make sense that more expression of a liver enzyme, of a lipoprotein enzyme with lower HDL levels. And then if you look in blood, actually the effect sizes swap. And so I think what's going on here is all the methods, including ours, is being disturbed by the number of weak instruments. And I'll show you a picture of that. But I just want to highlight the effect sizes go from negative and with other methods having pretty small intervals to positive with still small intervals. I think we're doing a better job by having larger intervals, because I think it's not plausible that the effect, given what we know about LibC, it's not that plausible that the effect would change from negative to positive. So this is an easy case. I'm actually going to skip over it. I think this is a case where everything works out pretty nicely. I want to focus on the problem because it's more of an interesting area for method development. So if we run our pipeline with all of the heuristics, including like how we selected instruments, we get a lot of instruments with weak effect sizes on the x-axis. And then you can see they have a lot of dispersion on the y-axis. And I think these are probably, these are problematic. I think we just need to do a better, more motivated approach to identifying instruments when this is in the case where this is eQTL gen, where the sample size is going up from 500 up to 30,000. I think that's what's driving the these probably too small intervals. And yeah. So let me see. I want to make sure we wrap up on time. So we also have applied this to neuropsychiatric traits, and yeah, I'm just going to jump ahead to this example. So we looked at post-mortem brain QTL from PsychENCODE, and we paired that up with two different neuropsychiatric GWAS, so schizophrenia and bipolar disorder. We screened using TWAS, and then here we used, for speed reasons primarily, our Bayesian model for colocalization is quite slow. We swapped in eCaviar. And so this allowed us to kind of use MRLocus, the slope estimation, in a kind of a way that we could process lots of loci. So here we were processing like hundreds of loci in less than an hour, whereas previously it would have been a bit slower. We've turned this into a pipeline so we can run it, and then we can compare our effect sizes, which are on the y-axis. These are the change of the log odds per standard deviation of gene expression, and compare that to the TWAS z-score. And they're in the same direction. I think that would be expected, but the effect sizes are very different. And here, everything is significant in terms of TWAS, but only a fraction of them are significant. If you look at the, here it's a kind of a Bayesian, like integral of the posterior distribution overlapping 0. So we get, it's a big specificity filter, I think, on TWAS. And that, yeah, so this is schizophrenia. This is now changing the x-axis to the TWMR effect size, and then ours on the y-axis. And, again, they're kind of similar. They're in a similar direction. But again, ours are kind of, we have a little bit of larger intervals. We're passing along, I think, more uncertainty. And then I think an important task is to kind of go in for each gene and do examine the data that's driving, these slope estimations, and observe both the uncertainty on the x-axis and in the y-axis, and consider, like, so the dispersion as well as our certainty on the slope. So I think looking at these plots is really important, even when you're scanning lots of loci. And then we did the same for bipolar disorder. There's a lot less sample size, so we have a lot fewer candidates passing through. But we can look at those as well. So this is my summary slide. I think what's, I don't think this is like the complete solution. I think there's a lot of interesting pieces, and we haven't tackled everything. But what we did add, I think, we tried a horseshoe prior for per signal colocalization that induces sparsity. I think that was interesting, but it's also a bit slow, and other methods work as well. We're often using conditional analysis in lieu of clumping for reasons I mentioned previously. we're explicitly modeling the instrument effect standard error. And I think this is a key aspect that we really need to account for that, because it's actually the x-axis where there's more uncertainty in these molecular QTL studies. And we elevated dispersion as a variable of interest in itself. So also other methods have kind of the, the other methods have used like the I-squared, like PTWAS outputs I-squared as a measure of the variability across signal clusters. And here we're modeling it with its own parameter. And of course, lots of things are missing here. And for example, we're not formally accounting for pleiotropy. We're just relying on having multiple cis variants in the same direction. So, we could probably do a better job formally accounting for that. Other methods consider multiple genes in a locus, and that's probably a good idea. Biology is complicated, and there could very well be multiple different mediating molecules in the same genomic locus. Other methods kind of model multiple tissues intrinsically, whereas we're kind of just hoping that we picked a good tissue. And as additionally, like I think really interesting now is to also tackle multiple levels. So to see how the mediated heritability passes through things like chromatin or methylation, through expression or splicing, and then into protein. That's, I think, beyond the scope of what we tried in this first pass. So we have an R package and a publication. We're really interested if anybody tries it out and has any kind of feedback or issues, please feel free to reach out to us. And I'll just acknowledge again the co-authors Anshi Zhu and Nana Matoba, Jason Stein, who's co-PI on this work, and our co-authors Emma, Amanda Yun, and Joe Ibrahim, and the funding. Okay, I'll stop there. Thanks, everyone.
Thanks, Mike, for this very fascinating talk and for taking your time to suffer the difference in the time zones, because it's early morning for you. So if anyone has any questions, can you please raise your hands or post them in the chat so I can read them out for Mike. So maybe I'll start. So actually, it's quite a naive questions, but from your experience, how do you pick the best tissue for a given trait or a gene? What's the best heuristic?
We've used, let me scroll back. We've used this paper previously. So this is a paper from the GTEx Consortium where they ran, I'm forgetting if this was TWAS, just straight TWAS, or if it was PTWAS, but across many, I think these are a lot UK Biobank traits and GWAS and the GTEx tissue clusters, right? So like here we have like lymphocytes and spleen, or brain and pituitary. And I think this is a good screening approach, and they've already done it. So you can kind of, if you're using GTEx as a panel, you can kind of see where do things light up, like what tissue is lighting up. But I don't have a better method than this.
Mm-hmm. And I have also another question about the LibC example.
Sure.
So in the plot we show that for the small effect as eQTLs, there's a lot of variation. So what do you think is going on here? Like, what's the biology underlying this?
I think everything is significant when you go towards 30,000 is a massive sample size, and there's just it's just going to be a lot easier for things to pass through, and the QTL, these association studies are, there's a lot of noise. There's potential like insufficient correction, potentially for kind of batch variation, or I think there's a million explanations for why a weak instrument could pass through, and I think we just need to do a better job in screening a way that I think overly filtering out signals is also a problem, because for a small study you would lose everything. We tried that. We like lowered the threshold for small sample size QTL study, And then you really start, if you lose, like here, 3 signals, if we lose one, we get a huge inducement on the induced error on the gene to trait effect, right? Each one really counts when there's only 100 or 200 samples. But then when we go to 3,000 or 30,000, then this is actually destroying the inference. So I think we need better things than heuristics to deal with this.
Mm-hmm. Yeah, thanks. Yeah, I totally agree. Does anyone have any questions? If not, okay, Tom, go ahead.
I'll ask a question first, and then the other Tom's got a question as well. So mine was obviously there's in terms of how you define the signal clusters, you've got an R² threshold, but also the reference panel and how good a fit it is to the particular data sets is going to be really important. And I just wondered whether you'd explored how sensitive it is to that.
That's a great question. Very important. And we have not, that was on our list for the fall, was to use the wrong reference panel for clumping. And in this, the simulation is fine, but we're actually using the same LD panel, both for generating the data and for using the clumping. So that's a little bit cheating. I think that's actually really important. And so so one thing is, I think we do have the ability in our software, in our method, to have different LD matrices for the QTL study and the GWAS. So, it's a generative model. So we're generating the observed marginal associations, and that's fine. But you do need the right LD panel. We should do a assessment where we swap in the wrong one and show how badly things go. And just to highlight, Yun Lee and her group have worked on a tool called TopLD, which is LD matrices derived from the TopMed consortium. And those seem to be much better than the LD panels that we use, just because when we started the paper, that resource wasn't available yet. But that's probably where we should go. Also, then I think area where we haven't solved this problem is when we have kind of multiple populations, and it's not a good idea probably to average the LD matrices across the multiple populations. So maybe you want to be doing, pretty complicated meta analysis within populations that have kind of basically don't average the LD matrices across populations, but you want to be splitting this out. It probably gets really complicated in terms of methods, and we haven't addressed that. Thanks.
Perfect. So I switch to Tom Palmer, who has a question.
Thanks very much, Mike. I was just, your comment about the sort of uncertainty, I think it was in the gene exposure estimate. So you'll notice in our software for IVW and for EGA, so we sort of give 3 choices. So there's first order weights, second order weights, and Wes Spiller in the radial stuff has the modified second order weights. So the second order weights there are the ones that incorporate the uncertainty in the genotype exposure associations. So you could have that in your IVW comparison if you wanted to as well.
Okay. Yeah, I knew, so this was it was a new effort, like a new space for me, and I'm sure I was missing things. So I should have, I'll make sure to point that out now that I know about it. So that would probably recover better inference here, because having those, passing that along would improve things here. It's likely it's happening. It's just I'm not passing that along, so it's not taking that into account. Okay, so good to know that it actually does exist in the IPW case.
Yeah, brilliant. Thanks.
Or I have a question. Do you have a sense for how often people are using that?
It's kind of gone in a loop, actually. So in the early days of MR, we used the second order weights almost all the time. And then when the 2 sample stuff became really popular and MREGA came out, there was just this kind of shift to using the first order weights a lot more. And that sort of remained the default, really. So yeah, we did used to do it about 10 years ago.
Yeah, I think, it was a new area for me when we started this, like 2019. 2018, no, 19. And then we, I was just trying to see how do people bring along the standard errors for the instruments and in the QTL study. And I just didn't see anybody actually using, like it would just, in terms of code, it would just not be used. And so I was thinking like, are we the only ones doing this? I know there are, you know, I think MRLink also makes use of the standard errors for the QTL study. But I think it's really important. I'm glad to know it's actually, you can do that in the IVW. Now I'll correct myself when I give presentations.
That's great.
So thanks everyone for the questions. So if you want to talk to Mike some more, you know, around possible collaborations and research interests, there's a link in the chat, and I also circulated the link earlier in the email I sent to you. And shall we take like a break for like 5 minutes and meet on the other link in around 2?
Great.
Thanks a lot, everyone.
Thanks for your attention, everyone.
Thanks. Bye.